by Dr. Timothy Henrich. So Dr. Henrich is Associate Professor at the University of California, San Francisco, and he specializes in infectious disease, HIV research, and translational immunology. His work focuses on HIV persistent cure strategies and the interference between viral reservoirs and host immunity. And he leads collaborative studies bridging clinical and basic science. And today he will be talking about a lentiviral-based gene-modified hematopoietic stem cell approach for HIV-1 cure that leads to partial protection of uninfected cells, but also lead to ongoing lentiviral RNA expression, which is a risk for potential persistent inflammation. So over to you, Dr. Timothy.
Well, thank you so much for the invitation. I'm sorry I can't be there in person. And I also apologize for the fairly lengthy title of the talk today, so I apologize about this. But I'll be talking about lentiviral-based gene modification and other gene therapy approaches for HIV cure with a focus on our AIDS Malignancy Consortium 097 trial using a one-tax lentiviral vector delivery to make hematopoietic stem cells resistant to HIV infection and the issues that have come up for us over time in the clinical setting with this approach. Just some disclosures. We'd like to just give a little bit of background, as many may not be retrovirologists in the crowd outside the lentiviral delivery community, but HIV is a relatively difficult virus to purge from viral reservoirs. And this is because it integrates into host human cells and essentially hides from immune surveillance as well as from antiretroviral therapy, which can suppress replication but is not able to touch the viral reservoir. So during early infection, you establish a pool of infected CD4 T cells, also myeloid cells as well, macrophages, macrophilia in the brain. And even after ART is initiated, many of those initial infected cells will die, but you do establish a pool of latent virus within tissues across the entire body. And this can be in organized lymphoid tissues. Actually, the gut mucosa is one of the largest sites of viral persistence over time. We also see this in bone marrow, the brain, spleen, and many other sites as well. So it persists across the body. But what's interesting is that in long-term ART, or antiretroviral therapy use, we actually see that less than 1 in a million of our resting CD4 T cells can actually harbor replication-competent HIV DNA or proviral DNA that's integrated into the virus. So when we're trying to look at curative strategies to eliminate these resistant reservoir cells, it's not looking for a needle in a haystack, it's looking for a piece of hay in a haystack, because these cells otherwise look normal to the human body as they have integrated DNA. They proliferate over time to persist, to establish this persistent reservoir. And ever since combination antiretroviral therapy was developed, rolled out clinically in 1996, people have been on antiretroviral therapy for decades. And despite this, when someone stops antiretroviral therapy, usually predictably within 1 to 2 or 3 weeks, we'll see rapid viral rebound with detectable HIV RNA in the plasma. This is actually one of the very first studies of treatment interruption, where people, after being suppressed for a certain amount of time on antiretroviral therapy, were stopped and virus rebounded quite quickly in almost all of the participants that were treated. And what's interesting is that 30 years later, we are seeing exactly, or actually almost 40 years later, we are seeing exactly the same rebound pattern, where if we do stop someone who's taking long-term suppressive ART, there's a predictable rebound suggesting that the viral reservoirs, although there is a slight decay over time, are quite persistent. And this is the challenge in order to purge these viral reservoirs and to protect cells from becoming infected over time. Just to show you a little bit about the viral life cycle, to show you that HIV has a surface envelope expression, GP120, this little ball on the outside of the viral virion particle that binds to CD4. This hence the CD4 T cells are one of the main reservoir sites of persistent infection, but it also needs to bind to another co-receptor. So this is either CXCR4 or CCR5 commonly. There are some other co-receptors that it can use under duress, but these are the two main ones. HIV then once binding to CD4 and a co-receptor, is brought in close contact with the membrane of the cell and delivers its internal genome. It's an RNA virus, but at this time it goes through uncoating of a capsid that's inside and reverse transcribes at the same time. So there's reverse transcription from RNA to DNA that uncoats and then integrates into the host cell genome. And during any type of immune activation, stress, antigen presentation, or response by these T cells, HIV genes can be turned on, transactivation and transcription and translation, and then viral processing to produce new virion particles. And you can also get mRNA activity and HIV RNAs produced even without proteins or even without virion production over time. And so we often see not just integrated DNA of people on ART, but we almost always see RNAs as well, intracellular RNAs, not the plasma necessarily in any sufficient quantity, but intracellular RNAs. Now, obviously, all of these different life cycles have been targets for potential gene therapy approaches. Obviously, if we think about entry, either inhibiting and or downregulating expression of co-receptors, obviously CD4 is important. We don't want to modify CD4. We're going to lose a huge amount of our immune repertoire, immune health, and that would be devastating for the person. But if people notice that people can live with homozygous mutations in CCR5, and without a functional CCR5, people seem to live healthy, long lives, et cetera. They don't seem to have any adverse health effects in any major terms. And so many of these approaches are now thinking about, can we reduce expression of this viral co-receptor, either with a zinc finger nuclease or small interfering RNAs, for example, and other techniques. But there are obviously many other different life cycles that we can target using gene therapy approaches for HIV cure. Right now, obviously, CRISPR-based gene modification is on the forefront of many of the studies at this meeting, but also in the HIV cure space. This can involve anything from modifying, again, human genes that code for co-receptors like CCR5 to targeting integrated DNA itself to create brand strakes or mutations that would prevent HIV from either becoming translationally active or producing infected virions. But obviously, to deliver a CRISPR system to a very rare human immune cell that persists across all the tissues in the body in very low density that looks very much like another uninfected cell is obviously the challenge. So although in vitro and in smaller animal systems, this has shown some promise, I think really the delivery method has been the main challenge. Because of that, we are actually still in our field going for the low-hanging fruit. And that, of course, is modifying the CCR5 co-receptor that HIV needs to enter the cell. There has been one clinical study to date that has had a kind of rigorous evaluation and also now subsequent studies in humans, which is gene editing CCR5 in autologous CD4 T cells of persons infected with HIV. And this was a seminal paper. It's now back from 2014, so over 10 years ago. And this is where we still are, unfortunately, in the technology of gene therapy in vivo in clinical studies for HIV. And what this approach did was use the zinc finger nucleases ex vivo. So people underwent leukapheresis. They harvested CD4 T cells. These were then modified with zinc finger nucleases to reduce expression of CCR5 on the surface of cells. These were then reinfused into the participants. And what you can see on the right top box is that actually, even though these are not hematopoietic stem cells, so these are adult CD4 T cells, differentiated CD4 T cells, they do have some stem cell memory-like properties, naive T cells, et cetera, that become modified and go in. But what they noticed that over time is there was an initial bump in the CD4 modified cells, and then those kind of waned, but they actually reached somewhat of a steady state that lasts up to about a year or so after infusion. So even though they're a minority of the total population of cells, that they do persist over time. And in the bottom panel, this is now showing CCR5 modified CD4 T cells in gut mucosa. Again, the gut mucosa is one of the main areas of viral persistence and viral activity even during acute infection in the Peyer's patches and the lymphoid structures within the gut. And so this was promising that these modified peripheral CD4 T cells could remain over time and persist. But do they help or do they protect? And they did a treatment interruption. If you look at the graph from my second slide, back from the early 1990s or mid-1990s that essentially when they stopped ART, or antiretroviral therapy, there was rapid recrudescence and rebound of virus in this space, and there also was not a huge bump in the amount of CD4 T cells with CCR5 modification. So actually there wasn't a huge amount of selection pressure in the setting of active viremia. Some of the set points of virus, that means that the peak viral load during rebound was slightly lower during the initial infection when compared to initial infection, but it was pretty modest. And ongoing studies now are using cyclophosphamide and other drugs to make space for these modified T cells to make more room, but we're still waiting for further data on how this is going to work in a larger cohorts going forward. Given the fact that we really haven't made a huge amount of in vivo progress with gene-modified therapies, now there are CAR T-cell studies now ongoing, including within our own group, so that is ongoing right now. But I wanted to talk a little bit about the AMC AIDS Malignancy Consortium 097 trial, which is using a one-tax lentiviral delivered combination gene therapy approach in autologous stem cell transplant patients. So people with HIV on suppressive antiretroviral therapy that need autologous stem cell transplant. So they're going to get CD34 hematopoietic cells harvested, they're going to get conditioning chemotherapy and those to reinfuse for hematologic malignancy. This was an opportunity during that time of after apheresis and collection of the CD3 stem cells is to modify them using a lentiviral vector. And so what was used was a one-tax, which is a third-generation lentiviral vector delivery approach. It's based on HIV-1, as in almost all of the lentiviral vectors that are used today, with the tax from HDLV involved. Also use an MNBU promoter to drive the genes of interest into these cells. In the AMC 097, give it a little bit more details about this vector and what we were doing in terms of gene modification. And so far, 11 participants, this is the initial pilot study is now complete, with HIV on ART requiring autologous stem cell transplant for hematologic malignancies have already been undergone this gene modification therapy technique. So what happened was following beam conditioning, patients received lentiviral delivered gene therapy to the CD34 positive enriched stem cell. And the one-tax vector actually was very clever. This was by Joe Anderson's group at UC Davis and Merdad Abedi as a clinical oncologist on the clinical side. But what the vector contains are 3 resistance genes. So it's not just targeting CCR5 like the Big Finger studies on peripheral CD4 T cell. This included a shRNA for CCR5, so inhibiting RNAs to reduce CCR5 expression, transport to the surface and expression. It also included a chimeric human macaque TRIM5 alpha gene. Now the TRIM5 alpha gene is a viral restriction factor in HIV that prevents uncoding and reverse transcription. And the human TRIM5 alpha is not very good at restricting HIV-1, and that's why we see HIV-1 as the main human lentiviral infection in the HIV family. Whereas the macaque version of TRIM5 alpha is actually very efficient at restricting capsid uncoding and viral reverse transcription of HIV. And so if you make a chimeric with the human and a macaque motif, you could actually gain the function of the macaque TRIM5 in the human gene. So this is actually a chimeric non-human primate / human gene that's inserted into the cells with a lentiviral vector. It also has an HIV TAR decoy. TAR is a transactivation element. It's a small hairpin RNA that actually helps initiate transcriptional transactivation in infected cells. And this essentially is a small RNA as well that acts as a decoy and essentially confuses HIV and prevents it from becoming transcriptionally active. So it's shutting down 3 different things. It's shutting down entry through CCR5 co-receptor expression that HIV needs to enter. It's reducing viral uncoding of the capsid and reverse transcription. And it's also preventing HIV transactivation and transcriptional activity. So even if it does manage to integrate, it has a hard time turning on to make new proteins and virions. This also has a truncated CD25. This is CD34 stem cells do not express a lot of CD25, and this was thought to be a good surface marker that they could engineer in order to enrich the CD34 modified stem cells for infusion. So you can identify those cells by CD25, sort those, and then you can inject as many or proportion of those as you like. Unfortunately, when these stem cells differentiate into lymphocytes, they start expressing CD25, so unfortunately it's not able to be used as an adequate marker of gene modification after differentiation into adult lymphocytes in vivo. So there are three cohorts in the study. Each got either a 1-to-1, a 5-to-1, or a 1-to-0 of modified to unmodified cells, so different ratios, or all modified cells. And just to show you some of the demographics here, these were mostly people under the age of 60, mostly men, and had non-Hodgkin's and Hodgkin's lymphoma, with one person having a plasmablastic lymphoma. So again, they had malignancies that required treatment. What was seen over time, now this is just showing the CD4 T cell counts after autologous stem cell transplant, and what we showed was a very similar trend that we see in our hematologic malignancy patients that receive autologous stem cell transplant without gene modification. So there's an additional dip in lag and lymphocyte reconstitution, but over time, the CD4 T cell populations do increase. And this is relatively typical for what we see during stem cell transplant, even without gene modified cells. Now, what's very interesting is what we had noticed in these participants is that there was actually quite a bit of persistence of mature lymphocytes, peripheral blood mononuclear cells that harbored the one-tax lentiviral vector. And this is just showing ratios, so where it says 0.16, it would be 16% of the total peripheral blood mononuclear cells would be harboring the lentiviral one-tax vector, suggesting that it's having activity within that cell. And you can see that in certain participants, and we had some dropout, there's about a 30 to 40% mortality rate in this cohort, but that's not related to the lentiviral vector. That's par for the course, unfortunately, for these advanced lymphomas in the setting of long-term HIV infection. But what we saw in certain individuals, that we were seeing up to 25, even 40% of the peripheral blood mononuclear cells in these participants carried the lentiviral vector over time. So these are not just CD4 T cells, but other lymphocytes, B cells, monocytes, myeloid, immune cells, et cetera. And so showing that this proportion can actually work quite well if you want persistence of stem cells over time. Now, maybe 40%, 50% may not be sufficient. I mean, there's been some modeling showing that if you really, really need to reduce the target for novel HIV, so if you need to reduce CD4 cells, it's probably going to be 90 to 95%. So this is lower than we might need to achieve full protection from de novo infection in vivo. What we also saw, and this is looking now across time on the x-axis and the participants on each of the rows, and then their group on the left on what the dose was in terms of hematopoietic stem cells, looking at their viral load. So this is looking at the RNA copies in the peripheral blood, in the plasma, in participants over time. And what you can see is normally we'd like to see this 40 or less. So our assays either have a 20 or a 40 cutoff for quantification of the viral load, and that would be considered fully suppressed on ART. And what we see over time is that most people actually remain virally suppressed, although we do see levels 100, 98, 79. And this was a bit confusing at first because we would not expect to see these low-level, what we would call low-level viremia in people that are stable on ART. And all of these participants remained on ART except for participant 3, right around day 183 due to access to care issues, was having issues with medication adherence. As you can see, had low-level viremia into the 2000s or so over time. But what was interesting, despite not taking ART, this participant never went above 2,000 or 3,000 copies of HIV, making them what we would call a controller. So whether that happened to be because of lentiviral gene delivery or not, we're still working on, but I can show you data that may or may not be the case. What's interesting is we also noticed that when you use many of the assays that we use to quantify HIV viral loads, so these are standard hospital-based tests using PCR or probe amplification methods, target conserved regions of HIV that are also still expressed on the proximal GAG and LTR junction of the one-tax lentiviral vector. And so some of these low-level blips, when you use another commercial assay that is farther downstream in the HIV GAG genomic region, these all disappear. And so these all become undetectable. So except for participant 3 during this time when he stopped ART. And so what we're seeing is that there is low-level detection of lentiviral RNAs being produced and released into the plasma in these participants that looks like it might be their HIV, but when you use an assay that is downstream, that has a genomic region that is not included in that lentiviral vector or packaging system, that you see that disappear. So that was the first important finding that we saw. We also had the opportunity to do a treatment interruption. So if we, in other words, our one participant, participant 10, after 2 years, after initial stem cell therapy and gene therapy, was doing well from a lymphoma approach, was in remission, and was fully suppressed when we look using the viral load assay that looks just at HIV-1. And this participant then underwent a planned treatment interruption. So the thought was to stop antiretroviral therapy and to watch and see if the virus rebounds. And if so, are the modified cells going to increase in frequency and control that rebound? And so what we saw is, again, using various different assays, the Roche viral load assay here on the left blue column actually detects some, actually cross-reacts with one-tax RNA vector. And on the right, the Abbott viral load actually does not. And what we saw is that, again, before ATI, there was this low-level detection with the commercial assay that does cross-react, but nothing with one that does not. But after therapy was stopped, we actually saw somewhat of a lag and then an increase in viral load copies up to 5 to 6 million, which is very high. But when using the viral load assay that is only for HIV was about 1.5 million, suggesting that a vast majority of RNAs that we're seeing in the plasma are actually coming from the lentiviral vector. During the time of acute inflammation, there's a lot of proliferation of cells. There's also a lot of cell death involved during acute viral rebound. So there's a lot of release of lentiviral RNAs into the plasma. What was interesting, though, is that when we saw that there was actually a transient increase from about 3% to up to 30% of modified stem cells during that initial viral rebound, suggesting there may have been selective pressure and some protection of those cells from the gene. The reservoirs did not change, but I just want to skip ahead to show you that the big question for us, and the last question, was, are gene-modified cells becoming infected with HIV? Are the ones that are harboring the one-tax lentiviral vector protected from HIV? So we developed a novel, high-throughput, relatively inexpensive assay using droplet digital PCR with encapsulating individual cells within in a lipid microenvironment. And then we performed duplex PCR, one for a portion of the one-tax lentiviral vector, this MNDU promoter, and also with HIV-1 that does not cross-react. And what we can do is if we see one, both, or the other, we can tell if this cell harbors lentiviral vector alone, HIV alone, or both, or none. And it's a high throughput method, and what we, this is just kind of showing the assay setup, the upper right-hand quadrant here. These would be HIV and lentiviral affected cells, and then in the other quadrants would be only lentiviral vector or only HIV-1. And what we saw using this assay is in participant 10 before and during different stages of viremia, that there was about a 75% protection of modification. So the modified stem cells were 75% less likely to be infected with HIV than cells that weren't. So it was not 100% protection, but it did confer at least partial protection to de novo infection during very high levels of HIV-1 replication. Now, I just wanted to finish up just to show that we learned a few things from this study. This was one of the very first lentiviral vector-delivered gene therapies for HIV. It also led to false positives in several kind of industry research standard HIV-1 DNA and RNA assays. So we have to be very careful, even when working or doing therapy studies with people without HIV, that this may actually trigger viral load testing to be positive in that setting. So this could cause a lot of anxiety, a lot of follow-up, and this needs to be addressed. That necessitated the development of new viral quantitation platforms that do not cross-react HIV with the one-tax lentiviral vectors. And many of the other lentiviral vectors, by the way, that are made for CAR T-cell development also will cross-react with these assays as well. We also saw that RNA expression of the lentiviral vectors increased dramatically during ATI, or treatment interruption, and during the tumor recurrence in other participants. And so that suggests that these vectors, although they're not replication competent, we looked, they did not recombine with HIV, they're not replicating, but they are making a lot of RNA, and that RNA is being released into the bloodstream. And so I just want to end with that. That's actually a little bit scary to us. The fact that you could actually have, under the setting of some type of immunologic immune activation or inflammation, a high level of transcriptional activity of these lentiviral vectors that's released into the periphery and in the circulation. And the reason for that is that we know that in the setting of HIV, so when you treat HIV, you suppress viral loads mostly, but you still, as I said before, you still have these viral RNAs that are being produced. You still will see very, very low level, maybe 1, 2, or 3 copies per ml of plasma as well, even on suppressive ART. And we know from recent data and recent research that's been presented that these RNAs can trigger immune activation. They can trigger TLR activation, and they basically can trigger immune activation, which can lead to inflammation over time. And so we are now investigating whether these lentiviral vectors, whether for CAR T cell modifications or for direct lentiviral delivery to either peripheral cells or to hematopoietic stem cells, are they going to lead to potential increased immune activation inflammation over time? Again, too early to tell. I do not want to be alarmist, but this is something that we are actively investigating and need to think about when using these types of approaches.
Dr. Henrich.
With that, I'd like to stop and thank everybody who did all the work. Sorry for going a little bit over.
Thank you, Dr. Henrich. So the session is open for one question, maybe.
I have a question. So in the treatment of HIV-infected patients, what is your thought process on using a retroviral vector instead of a lentiviral vector? I would have thought retroviral vector would have been a normal choice, right? And then the second question is, have you done any quantitative viral outgrowth assays on the other patients? And what is your decision to do the ATI? Why not do a QO before doing the ATI?
Oh, great question. So I'll clarify, these are retroviral-based vectors within that. So they are HIV-1 based. They also have pieces, I mean, they have the TAX gene from HTLV1, but mostly these are HIV-1 based, right? So they're retroviral vectors in what they're being used. And so, and that's again why we're seeing a lot of the cross-reaction with the HIV test as well, if that makes sense. In terms of the deciding whether to do the ATI, so there's been a lot of controversy and or discussion, not controversy, but discussion about the safety of doing treatment interruptions in the setting of HIV. This is actually something that we do quite commonly in the HIV cure space. We're doing these in the US, Europe, now in South Africa, for example. So these are becoming more common. They're actually shown to be actually quite safe during as long as you kind of follow them closely, as you start to see this, you can restart ART, those viral loads come down. So there's a lot of evidence now that's safe. We don't want to do this in everyone. We want to make sure that the tumor was in full remission. We wanted to make sure that they were fully suppressed on ART. Their CD4 T cell counts had reconstituted to pre-transplant levels. So we were looking at, that's why we only did it on one participant, unfortunately, for this study.
And
I think you had another part, but I can't remember I'm sorry. The
QOO assays. Have you done quantitative viral outgrowth assays on?
Yeah, so we've done. So we have done viral load assays. We are able to detect replication competent virus in pretty much all the patients that we've tested. There's another assay called an intact proviral DNA assay that people who do a lot of HIV reverse are familiar with, the IPDA. Unfortunately, the IPDA interacts with the one-tax vector. As well. And so it's unfortunately we can't use that shortcut to look for replication or intact proviral DNA as a surrogate for viral. Yeah.
So can we have one question, quick answer? Yes.
Okay, so I just have one quick question. Are people still using TAT, viral TAT? Because years ago when I was a postdoc, we used to have TAT and REV as one as a regulatory plasmid, apart from GAG pollen. And so are still people using TAT as part of their regulatory plasmid?
Not that I'm aware of. I mean, TAT TAT has a lot of problems. That TAT itself is quite immunostimulatory to cells. Right. So in fact, it actually can cross the membrane. So if you just put TAT into a viral culture system, you can activate those cells and you can cause, or even with not a culture system, but if you just put TAT in to CD4 T cells or PBMCs, you're going to get a certain amount of immune activation involved as well. And so I think people are trying to kind of avoid TAT-mediated potential cell toxicities in some of the constructs as they go forward. I think the problem is they're doing a lot of deletions within these vectors, but not necessarily removing entire gene sets. And so that's why, for example, there's no envelope expressed in any way of HIV in any of these vectors, native envelope, after integration, but there's still a huge amount of envelope DNA sequence that's left over that we then detect using various assays as well.
So we quickly switched to TAT-less regulatory plasmid long back, like way back in 2009 or 10, probably before I came back. So I just wanted to know people
again, for the reason that TAT is not.
Yeah, I think it's superfluous for expression, so I think one can just deal without that. Thanks a lot.
Yeah.
Nice talk.
Yeah, great question. Yeah.
So I think there's one more question.
Sir, I have a basic doubt. After lentiviral-based gene modification, can you characterize the clones, edited clones, whether you come across any mutagenesis or transgene silencing, those kind of things you come across? Any safety studies you have done so far?
Yeah, that's a great question. So when we did notice that there was an increase in the vector RNA that we were detecting in the blood, especially in this one participant that had tumor recurrence, and there was the thought that there could have been mutagenesis, clonal expansion of of the vector containing cells, because we know that some of the CAR T studies that are coming out, et cetera, things like that. Wanted to make sure. So those studies were done, and there was no evidence that there was clonal expansion of cells that harbored vector, and so we don't think that was leading to clonal expansion of cells, or is that what you're asking? Just want to make sure.
Okay, thank you, sir. Thank you very much.
Yep.